What is the link between immunity and nutrition? Have you often wondered? I'll try to attempt to answer this question in my presentation today. My name is Dr. Ebenezer Satyaraj. I'm Director of Molecular Nutrition at Nestle Purina Research in St. Louis, Missouri. Me and my team try to understand the relationship between diet and health at the molecular level. I'm an immunologist by training. I've divided my talk into two parts. In the first part, I want to attempt to answer two important questions. And the questions are, why enhance immune health in a healthy animal? And second, how can you do that through food? And in the last part of my talk, I'll walk you through two examples of studies that we conducted demonstrating the immune benefit of adding bovine colostrum to diets. So let's dive in. I titled the slide Immune System, 3 F's and 2 Q's. And we'll come back and tell you what this 3 F's and 2 Q's mean. What I show you here is the overview of the various cells and processes that are involved in initiating an immune response to a challenge. What you're seeing here are phagocytes responding to a bacteria or a viral pathogen, processing it. Depending upon the type of antigen and the pathogen, it is either presented to the CD4 T cell or a CD8 T cell. There is release of cytokines, proliferation of T cells, activation of B cell, and generation of an antibody response. In the case of a viral pathogen, you have the activation of CD8 T cells and a cytotoxic T cell response. It is fascinating to think that in our bodies and in the bodies of our pets, these processes are happening 24/7, because of which we are able to lead healthy lives. But if you really boil down to what are the key things that the immune system does for us and our pets, and how does it carry out those important functions, it really comes down to 3 things as the primary role for the immune system. The first is to keep the bad stuff out. This is the barrier function. The second is to check if anything has gotten through. This is the surveillance function. And then finally, how do you deal with the intruder? This is the defense function. So fundamentally, these are the 3 primary functions that the immune system does for us. And it does it by asking 2 important questions. The first question is this me? Which is really the heart of self versus non-self. And if the answer is, if this is not me, what I'm going to do about it. So now we go back to what the 3 F's and 2 Q mean. 3 F's are the 3 primary functions that the immune system does for us. And it does that by asking 2 fundamental questions. Is it me? And if it's not me, what am I going to do about it? Let's answer the 2 questions that we started off with. Why enhance immune health in an otherwise healthy animal? Now, I've had other presentations that I've given to various groups where we get into the details of why and how immune response gets lowered in certain stages in life. But for the sake of time, I'm just going to give you the key highlights. And those are malnutrition, life stage, whether young and old, as well as stress can lower the immune status in humans as well as in pets. And when you talk about malnutrition, you also need to keep in mind this is not just undernutrition, this is also overnutrition and the lack of critical key nutrients like vitamins and minerals. And the fundamental reasons why immune system gets depressed or less effective in these stages revolves around 2 components. One, the ability to mount an effective immune response, and that relates to deficiency of certain cell types such as macrophages. And the second, on the other side, the ability to manage inflammation and prevent runaway inflammation or inflammaging that happens as dogs and cats and humans age. The fundamental idea is maintaining homeostasis and revolves around the role of an important cell type called as regulatory T cells. So let's move on to the next question that we'd like to answer, which is how can food influence the immune system? To be able to understand this, I really want to present two important ideas that are very, very key to understanding the relationship between diet and the immune system. The first is that over 65% of all immune cells in the body are present in the gut, organized as gut-associated lymphoid tissue, also called as GALT. And therefore, what we eat and feed our pets interacts with a significant part of the immune system. I'm going to take a little bit of time to walk you through this important slide because it is important to remember when you talk about diet and gut-associated lymphoid tissue, an important component to keep in mind is the role that the gut microbiome plays in not only enhancing, activating an immune response, but also educating the immune system. And you're really looking at a relationship between diet, microbiota, and immunity. So if you look at diet on your top left, what you see is diet provides important components to the microbiota. These include vitamins, fats, minerals, sugar, and importantly, fiber. Microbiota breaks these fiber down and releases short-chain fatty acids, which are important for cellular nutrition. Diet has an important impact on the immune system because it provides important components like vitamins, minerals, as well as certain key components of diets, which include toll receptor ligands, AHR ligands, NRF2 activators, all important components of activating and stimulating the immune system in the GI tract. And then microbiota also provides short-chain metabolites, and metabolites like short-chain fatty acids, as well as some of these components like toll receptor ligands to the immune system in the GI tract to be able to activate it. And the immune system in the GI tract, the GALT, in return, uses various agents like immunoglobulins, reactive oxygen species, etc., to see that the gut microbiota stays within bounds and doesn't invade the organism. Now, this is a very important tripartite relationship that is the basis of how diet impacts the immune system. Another way to understand the relationship between nutrition and immunity is to start considering that at 4 stages. So in stage 1, you're providing complete and balanced nutrition to the pet or the human. Now, while we take complete and balanced nutrition for granted, it is important to remember that when we don't have important components coming through diet, you start having significant deficiencies in the immune system, as you can see, for example, in war situations or famine. The second stage then is to optimize certain key micro and macronutrients, such as vitamins, minerals, and proteins. And together, both these approaches provide components to the immune system, but it is still passive. What is important and interesting is stage 3 and stage 4, when you're actively interacting with the immune system, trying to immunomodulate it. And we will see 2 examples of that in my talk today. Stage 4 is a very unique approach where you're trying to address a specific challenge that exists in the GI tract. For example, trying to reduce inflammation in certain disease conditions using an active approach of managing inflammation in the GI tract. So just to build out the stage 3 approach, there are kind of 4 ways of doing that. One is to be able to enable the efficient macrophage function by targeting innate immune receptors in the GI tract. And these includes a whole bunch of immune modulators that come from the microbiome, such as beta-glucans, mannins, algae, probiotics, and certain immunomodulatory components such as bovine colostrum that we are talking about today. Managing inflammation using DHA and omega-3 fatty acids, leveraging certain components of the diet, such as ARH, using our ARH pathway or NRF2 pathway also falls into this category. And of course, short-chain fatty acids produced by prebiotics, broken down by microbiome, are important supporting components for the regulatory T-cell development. And I mentioned earlier, these cell types are important in managing inflammation. Now, having done that, let's quickly walk you through the two examples that I was referring to. So first study that we'll cover is to walk you through a study that was conducted to demonstrate the immune and gut health benefits of bovine colostrum addition to diets in dogs. Here's a quick overview of the study. 24 Alaskan sled dogs between the ages of 2 to 6. There were 2 diets, 12 per group, control and test. 40 weeks that were conducted in our facilities in Alaska, and various immune and gut health measures monitoring during the study. And those measures include evaluating local immune response by measuring fecal IgA, evaluating systemic immune response by measuring response to canine distemper virus vaccine. Now, vaccine response is often a way that immunologists use to be able to understand the immune status of an individual, because you're really exposing the individual to a very innocuous, safe challenge. And you ask the question, how do they respond back? And then we also measured gut health parameters by looking at gut microflora profiles, and I'll quickly walk you through those results. So here's a schematic of the study. All dogs were enrolled in the study and fed a controlled diet during the pre-test phase, which lasted 8 weeks. Dogs then were switched to the test diet, and all dogs were given a canine distemper virus vaccine. The study lasted for 40 weeks, and we had monthly samplings collected throughout the study. Here you're looking at data of the fecal immunoglobulin that were measured in these dogs. And what you can clearly see is we compare when they all started in July to when the study ended in April, you can clearly see a significant increase in the amount of fecal IgA produced in the dogs that were fed colostrum supplemented diet, demonstrating that animals fed a diet with colostrum had enhanced local immune status to promote intestinal health. If you look at vaccine response to canine distemper virus vaccine, you're seeing exactly the same scenario. What you're seeing is fold change, and both the groups started at 1 during in July. And as time progressed, you see the booster response seen in both groups, but the group that was fed the colostrum diet maintained those titers almost until the end of the trial, suggesting that canine distemper virus response or systemic immune response is stronger and lasts longer in the dogs that were fed the colostrum diet. To evaluate the effect of stress on the gut microflora, we used an exercise model, and the model is described in the schematic. All these dogs have regular exercise, and the first thing we did was to break that exercise and give them a 2-day rest. And after that, dogs were put back onto the exercise schedule, but we collected fecal samples before and at multiple time points during the exercise. And the question we were asking is, does this normal exercise-related stress change the microflora in the GI tract? And this is an important component because once these changes go beyond a certain point, you start having situations like diarrhea in dogs as well as in cats. So this is a very safe way for us to be able to evaluate how strong is your immune system in trying to protect the changes that might happen in the microflora following a stress. And the results were quite surprising and quite nice to see that if you compare the before exercise stress versus after exercise stress patterns, the control group, there was a significant change in the microflora pattern, but in the colostrum-fed group, there was only about an 18% difference. So there was a clear stability of microflora that we could demonstrate in the dogs that were fed colostrum, showing the ability of these dogs to be able to handle stress much better. Quickly moving on to the colostrum study with kittens, again, very similar outline, 24 kittens, ages 8 weeks when they were recruited for the study, 2 diets, a control diet and a test diet. Studies lasted for 44 weeks at our facilities in St. Joseph, Missouri. And again, various immune and gut health measures monitored during the study that are listed in this slide. For immune analysis, we also measured fecal IgA. And for systemic immune response, we measured response to the rabies virus vaccine. And we used, again, a very similar but a different model in this case that I'll walk you through to ask the question, did they have a much more stable microflora? Again, a schematic of the study, 8 weeks pre-test period where all the kittens were fed the control diet. They were switched on, the test group was switched on to the test diet. We had the first rabies virus shot that was administered to all the kittens in the study. And a booster shot was administered somewhere at 37 weeks of age of the child. And then we evaluated the vaccine response in both groups. And just like what we saw with the dog study, what you're seeing again is a clearly higher levels of fecal immunoglobulin in the cats or kittens that were fed a diet containing bovine colostrum, showing that colostrum enhanced local immune status to promote intestinal health. If you look at the vaccine response, we had a slightly different pattern, but nevertheless, kittens that were fed the bovine colostrum diet had significantly higher titers at week 40 of following the booster shot, also showing enhanced systemic immune response. We evaluate the effect of stress using a slightly different stress model in this case, and the stress that we used was a vet visit. So during that 44-week of the study, all these kittens had regular vet visits. And we used that as an opportunity to ask the question, does that stress change the microflora? And are the kittens that were fed a colostrum diet able to handle that stress better? So right before every vet visit, we collected a fecal sample before and multiple times after, and did an analysis of the microflora pattern and asked the question, are there changes before and after the stress? And the results are shown here. What you're seeing here is, if you look at the kittens that were fed a control diet, there was a clear change in the microflora patterns before and after the vet visit. But the kittens that were fed the bovine colostrum diet, there was no statistically different changes seen, clearly demonstrating that, again, kittens that were fed a diet containing colostrum had a greater microflora stability under stress. So putting it all together, what both these studies clearly demonstrate that colostrum enhanced systemic immune response by the vaccine response being stronger and lasting longer. Colostrum enhanced local immunity by the higher fecal immunoglobulin that were measured in both studies, which is a marker of a healthy and active gut-associated lymphoid tissue. And then colostrum helped both the dogs and the cats handle stress better once they were on the diet. Clearly, immune status equals healthy animals. Both these studies have been published. The dog study was published in the British Journal of Nutrition, and the cat study or kitten study was published in the Frontiers of Veterinary Sciences. Thank you very much for your attention.